Hello, my name is Steve Kane, and I'm the Director of Product Management for NuGEN Technologies. Thank you very much for listening to the following webinar, where I'm going to discuss a NuGEN technology that can be used for targeted transcript sequence reduction in an RNA-Seq sample preparation workflow based on a technology that we've termed InDA-C. So to begin with, the InDA-C term, or insert-dependent adapter cleavage, is a technology that NuGEN's developed for enriching for desired transcript sequences in an RNA-Seq workflow following library construction. So what's unique about the mechanism by which the InDA-C approach takes place is we deplete transcripts that are not desired in the final library after library construction. So there is no interference with the original total RNA population, as would be the case with different types of hybridization capture methods. The specificity of this workflow relies on the design of specific InDA-C primers, which I'll describe in workflow slides in a moment. And what's important about this method is these InDA-C primers have a high degree of flexibility. They can be designed to target virtually any class of transcripts that you would like to remove from your library across a broad range of species. So for example, InDA-C can be used to reduce ribosomal RNA levels, but also different categories of transcripts which may also be abundant in your sample and not desired in the final library or the final data set. So by way of example, I'm going to show you some work that we've done with human blood transcript reduction, in particular to target reduction of human globin transcripts as well as ribosomal RNA. And this pie chart figure from a paper published in 2012 really illustrates the problem, if you will. So in adult human blood, the hemoglobin isotypes shown here, hemoglobin alpha 1, beta, alpha 2, and so forth, really comprise about 60% of the total RNA in a typical blood sample. If you add to this the ribosomal RNA levels, it presents a problem in developing an RNA-Seq sample preparation workflow where a large portion of your resulting sequencing reads would be dedicated to transcripts that you're likely not as interested in. These non-informative transcripts, if you will, comprised of globin as well as ribosomal RNA. So we've designed InDA-C primers to target the different adult human hemoglobin transcript types in an effort to reduce the prevalence of these messages in the final library. So let me take you through the RNA-Seq workflow. In particular, I'll emphasize how the InDA-C technology operates. So across the top of this diagram, you have the overall workflow, which begins with the input of total RNA through to the construction of a library that's ready to be sequenced on any of the Illumina platforms. In the initial first strand reaction, we hybridize a random set of primers along with a little bit of oligo-DT to conduct the first strand synthesis. So by way of example, we're showing the annealing of a single primer, but keep in mind that this would be random hexamers annealing along the length of the RNA transcript. We conduct the first strand reaction to produce an antisense transcript. In the second strand reaction, we incorporate a degradable nucleotide analog shown by these red stars, and then carry out the second strand reaction with DNA polymerase to produce a sense strand that's now labeled with these degradable nucleotide analogs. We will utilize the incorporation of these analogs in a subsequent step to carry about degradation that allows us to produce stranded libraries, or libraries that would give you strand-specific RNA-Seq data. There is an optional fragmentation step. We recommend the use of a mechanical fragmentation using a Cavaris instrument or an equivalent. It's optional because you can implement this workflow in an automated fashion where fragmentation is not required. So after the fragmentation step, we have molecules that are now double-stranded. And then we carry out the adaptor ligation to place adapters on that will allow for cluster formation and sequencing on Illumina platforms. The ligation that we perform is a blunt-end ligation such that when you carry out the adaptor ligation with both the forward and reverse adapter, there's two possible orientations shown here on the left and on the right. What we've done is we've labeled one strand of the forward adapter with the same degradable nucleotide analog, shown here and here, such that when we carry out base excision, that is cleavage at this degradable nucleotide point and final denaturation, we're left with only one form of the library that has both an intact forward and reverse adapter. This is then the only form of the library molecules that can be expanded during cluster formation and ultimately sequenced. So this is how we bring about a strand retention capability in the library construction workflow. And now comes the InDA-C step, or insert-dependent adapter cleavage. So at this point, you have molecules with intact forward and reverse adapters, and you have a range of different types of molecules as shown here. So you have blue inserts, which would represent different types of informative sequences that you'd like to collect RNA-Seq data from, but you also have different forms of libraries that have ribosomal RNA, or perhaps other unwanted transcripts that you would like to minimize in the final sequencing library. And we do this by using the InDA-C approach. So the way this works is we design gene-specific InDA-C primers that will anneal to regions of unwanted transcripts. Shown here, after the annealing event, we then extend from these InDA-C primers into the reverse adapter sequence, thereby forming a double-stranded cleavage site in the reverse adapter, shown here in these two black triangles. We then specifically cleave at this point, thereby removing the reverse adapter sequence from molecules such as ribosomal RNA or other unwanted transcripts and minimizing their presence in the final library. So you have a targeted depleted population that's been enriched in desired informative transcripts. We then use PCR primers that are designed to both the forward and reverse adapter sequences to expand the library. And the outcome then is a sense strand library that's been depleted of unwanted transcripts that's ready to be quantified and ready to be sequenced. So again, let me illustrate how this workflow can be applied first to a human blood sample and how we've used the InDA-C approach to minimize levels of ribosomal RNA as well as Globin transcripts. So shown here are results from 4 different normal human adults, and in this workflow we obtained blood using the PAXgene technology, which is a common approach for stabilizing and then purifying total RNA. And what we've done then is we've taken total RNA from these 4 different PAXgene blood draws, and we've prepared sequencing libraries either in the absence of InDA-C, where we carried out the workflow that I've just shown you and substituted water for the InDA-C primer step, or in the presence of InDA-C on the bottom. And what you can see shown in these yellow portions is that through the use of InDA-C, we've been able to substantially enrich for informative sequencing reads, or reads which would represent non-ribosomal RNA and non-Globin transcripts. Conversely, we've brought about a significant reduction in the percentage of ribosomal RNA reads shown in blue, as well as the Globin transcript shown in red. If you look at the sequencing reads, and these are 4 different IGV tracks showing the specific sequencing reads that are found in the 4 different Globin transcript types that we've targeted. In each case on the top, you can see the sequencing reads that are going to, in this case, the HBB isotype sequencing reads in the absence of InDA-C, and we've eliminated these in the presence of InDA-C, shown on the bottom. So by doing this type of reaction, then, it's really enabling one important category of clinical samples, namely human whole blood, to really get the most out of your sequencing resources. So you're now devoting the sequencing reads not to Globin or ribosomal RNA, but to other RNA types which may be more informative for your research study. One important aspect of this approach, because you are targeting the transcripts of interest, in this case Globin or ribosomal RNA, you want to make sure that there are no off-target effects, or they're not impacting other transcript types that you'd be interested to study. So here are results from the same 4 PAXgene draws Let's just look at sample 1, either in the absence of InDA-C or in the presence of InDA-C, we have a high degree of concordance in FPKM plots, or the representation, if you will, of non-ribosomal RNA non-Globin transcripts in each sample does not appear to be perturbed significantly by the use of these InDA-C primers. So there's a good specificity in this approach for targeting the desired transcripts. We also looked at the concordance of differential expression. We were looking at differential expression between a brain reference sample and a universal human reference sample shown on the bottom as UHR. So using 10 nanograms of total RNA input in each case, we are able to develop differential expression results that have a high degree of concordance to TaqMan assays. We've also developed a form of this product that's specific for work with human FFPE samples. FFPE stands for formalin-fixed paraffin-embedded samples. It's a very common approach for preserving and stabilizing human samples, in particular for cancer research. The results in this table represent data from our RNA-Seq analysis pipeline. We've generated these results with FFPE samples from human colon samples from 6 different patients. These samples range in age from 8 to 12 years and have RIN scores between 2.1 and 2.5. RIN score is generally an indicator of how degraded the sample is, which is typical with FFPE material. The ribosomal RNA as well as other RNAs are often highly degraded in these samples, and so it can be a challenge to conduct sample preparation for RNA-Seq analysis. In terms of input amounts, we also compared the FFPE samples to a human brain reference. The amount of total RNA used for the brain sample was 10 nanograms. For each of the FFPE samples, 50 nanograms. So let me just highlight some of the key results from this table. Looking at the percentage of overall aligned sequencing reads, they range from the high 90s down to as low as 71%. Typically, there is a broad range of performance across different FFPE samples because they tend to have a broad range of degradation as well as artifacts that are present due to the FFPE fixation process. If you look at the levels of ribosomal RNA, and this is total ribosomal RNA being the sum of cytoplasmic as well as mitochondrial ribosomal RNAs, for the brain control, we have levels of about 11%, and then there's a range of depletion with the different FFPE samples. And then lastly, the strand retention data. So in this case, we're looking at the percentage of RefSeq strand retention for coding sequences is in excess of 90% in every case and approaching 99% in several cases. We also looked at the ability to detect different genes from these FFPE samples using the RNA-Seq workflow that utilizes InDA-C, and that's shown here for these same samples. Looking at the transcript counts as a function of different FPKM thresholds. So shown in blue are the detection of genes with an FPKM threshold of 1 for brain, and then for the various FFPE samples going across. So we're able to detect in excess of 10,000 RefSeq genes in every case. If you decrease the FPKM threshold, you have a little bit higher level of detection. So generally very good results, very similar to what you would see with a fresh sample, in this case the human brain reference. We're able to extract very valuable biological data from these different FFPE samples. So those are a couple of examples of how we've applied the InDA-C technology to human samples. We've also had the ability to use this approach for a range of different model organisms, different species, as well as using InDA-C to target a range of different transcript types. And the way we've made this technology available to researchers for non-human species, if you will, is through the development of the Ovation universal RNA-Seq system. So if you remember the workflow that I illustrated in earlier slides, what this system provides is essentially all the reagents, all the enzymology required to produce RNA-Seq libraries, with the exception of the InDA-C primers themselves. So NuGEN provides a no-cost design service where we will work with researchers to design specific InDA-C probes to the transcripts of interest as well as the species of interest for your research. This table just illustrates a few examples of projects that we've worked on with researchers using this universal RNA-Seq approach. So for example, again, back to human, we've had researchers that are working with RNA-Seq studies from human cardiac tissue, where there was a desire to deplete levels of actin and myosin transcripts. In the case of mouse, we've developed InDA-C Primers to both ribosomal RNA as well as Globin. This is an interesting project with goat. In this case, it was work that was being done related to the cheese industry, the production of cheese as a food source. Turns out, in this case, they were working with goat mammary gland samples where the casein proteins, which are milk proteins, transcripts to these different proteins are highly prevalent, as is ribosomal RNA. So by depleting the levels of the casein proteins as well as ribosomal RNA, these researchers were able to heighten the levels of informative transcripts in their libraries. And then so on, a range of different examples, yeast, one example from plants, work that we've done with grape to target ribosomal RNA as well as chloroplast RNA, Drosophila, and I'll show you some data for Drosophila in a moment. These last two examples represent a different category where InDA-C can be provided, which is generally in the vein of a sort of host pathogen type model. So for example, this project with plant fungus was looking at a plant fungal species that infected barley plants. So the researchers depleted the top 30 expressed transcripts from the host barley plant in order to elevate levels of fungal transcripts for study by RNA-Seq. In a similar fashion with a human scenario, we've worked with a researcher studying tuberculosis to deplete ribosomal RNA in human, tuberculosis ribosomal RNA, as well as other housekeeping transcripts to elevate the levels of informative tuberculosis-derived transcripts in the final sequencing library. So the point that I really want to make from this slide is that the application of InDA-C is quite flexible, both in terms of the species that it can be utilized for, as well as a range of transcript types that can be removed from the final sequencing library. Here's some additional examples from products that we have commercialized for our different model organisms, and these are different examples of where we've targeted ribosomal RNA for depletion in mouse, rat, and Drosophila sequences. So if you look along this column, this is a similar workflow, once again, where we've either implemented an InDA-C primer design, or in the negative control, we've used water in the place of InDA-C. In every case, we get very good alignment scores. These are all fresh frozen samples, as shown on the bottom. The input is using 10 nanograms of total RNA from either a whole mouse universal reference, from a rat brain sample, or from Drosophila S2 cells in culture. So again, going back to the table then, the total aligned sequencing reads are very good, over 90% in most cases. And then look at the impact of the InDA-C primers on the percentage of ribosomal RNA. So we've been able to use this approach to reduce, for example, in mouse, the percentage of ribosomal RNA in the control sample from 80% down to about 8.7%. Very significant reduction in rat from 53% down to 2.5%. And then in Drosophila from 85% down to 7.6%. Very good strand retention, in this case approaching, 99% in every case. Shown here is a percentage of RefSeq exon reads, so sequencing reads that were going to the exon sequences, typically around 50% or higher in most cases. Remember, this is a total RNA sample, so you are getting information for both coding and non-coding RNAs. So there is a significant portion of sequencing reads which would still be going to either intergenic or intronic sequences. And then in this last column, again, is the number of transcripts that were detected. This is data similar to what I had shown you for the human FFPE sample. But what's revealing here is that in each case, by removing this large burden of ribosomal RNA, so let's take, for example, the mouse example, you're able to get a higher level of gene count data. So the number of transcripts detected is elevated by removing this background of ribosomal RNA in each case. So in each case, in the presence of InDA-C, the number of transcripts detected is elevated. Here's an example that shows you the reproducibility across, in this case, the Drosophila studies. So these are our 5 replicate cultures of Drosophila S2 cells. Again, the same workflow. We've used 10 nanograms of total RNA isolated from these cells, and then input to the InDA-C workflow. In this case, the product name is the Ovation Drosophila RNA-Seq system. And you're looking at the percentage of reads that are aligning to ribosomal RNA. Shown in blue is the absence of InDA-C, and then in the gold yellow bar, you can see the level of depletion that we see in the presence of InDA-C. So the mean across these 5 cultures, about 6.5%. So going from a percentage of ribosomal RNA in excess of 80% in every case, and reducing it down to an average of 6.5%. Turning to a plant system, and NuGEN has developed a dedicated product for work with Arabidopsis. In this case, for plants, you have the challenge of both ribosomal RNA, but also chloroplast ribosomal RNA, which can represent a large percentage of the library. So I've kind of turned things around a bit from the prior figures, and I'm showing you here the percentage of informative reads for a range of different library types. So in the absence of InDA-C, we have less than 10% of the sequencing reads would go to non-ribosomal and non-chloroplast ribosomal RNA. You can see the impact of using InDA-C primers to ribosomal RNA alone or to chloroplast ribosomal RNA alone. And on the top portion of this figure, you see that when we use InDA-C primers targeting both chloroplast as well as ribosomal RNA, we have a significant enrichment of informative transcripts in excess of 50%, either with 10 nanograms of total RNA or with 100 nanograms. So this represents a significant enrichment in the sequencing reads, which would be derived from the more informative transcripts in the library. So I'll conclude this webinar then by showing you some of the product names that incorporate the InDA-C technology. Each of these use the same overall workflow that I've described, from the input of total RNA through to the construction of a sequencing library that's ready to be quantified and sequenced on the Illumina system. So we do have complete solutions for human samples. The Ovation human blood RNA-Seq system utilizes InDA-C primers to both human ribosomal RNA as well as Globin. The Ovation Human FFPE RNA-Seq system is designed for either fresh tissue or FFPE-derived tissue and targets ribosomal RNA for depletion. And you can see these 4 different model organisms, which I've shown you data for, the mouse, rat, Drosophila, and Arabidopsis products. Lastly, something that I have not discussed today, but just to mention another product that's in NuGEN's RNA-Seq sample preparation workflow, the Ovation single-cell RNA-Seq system is utilized for low-input studies with as little as 10 picograms of purified total RNA or direct cell lysates. So with that, I will thank you very much for listening to this webinar. If you would like information about any of the products that I've described, as well as any other NuGEN products, please contact our customer service organization at the indicated email, and I thank you very much for your attention.